Ever felt like you're missing a crucial piece of the puzzle in peptide research? Don't let vital discoveries slip by. I'm Kai Rivera, Chief Investigative Scribe at Peptides.today, and I'm here to demystify the molecular world. Today, we're diving deep into peptides, proteins, and enzymes, the unsung heroes of biological systems. Get ready to have your research perspective expanded and your questions answered. Let's excavate some buried treasures. Many researchers often wonder about peptides versus proteins versus enzymes. These terms are everywhere in biological studies, but how exactly do they differ? And why do these distinctions matter so much for scientific discovery? Think of our biological systems as a bustling molecular city. Countless reactions occur every second. Peptides, proteins, and enzymes are the tiny, specialized workers that power this entire operation. Understanding these molecules helps clarify how compounds interact within biological systems and guides biotech research. First, let's simplify. These categories are not entirely separate. They actually overlap significantly. Proteins are generally large molecules, the complex heavyweights, all constructed from amino acids. Peptides are shorter chains of these same amino acids, smaller members of the molecular family. And enzymes, these are typically proteins, but their unique role is to accelerate chemical reactions. So think of proteins as the big family, peptides as the smaller members, and enzymes as specialized workers. Proteins are the fundamental building blocks and functional powerhouses of nearly all known life. From cellular structures to signaling components, proteins are everywhere in biological systems. These complex molecules are built from amino acids, like beads on a very long, intricate string. This chain then folds into a precise 3D shape. This shape dictates its biological function. The genome contains thousands of protein-coding genes, but these can yield hundreds of thousands of proteins. This enormous molecular toolkit performs diverse functions, essential for all known biological activity. Exploring these molecular machines is a major focus in current biotechnology and scientific discovery. Isn't it fascinating how intricately everything is connected at the molecular level? We're just scratching the surface here. This kind of deep dive into the, "Whoa, how did I miss that?" moments is what we live for. For more exclusive insights, breaking research news, and behind-the-scenes glimpses into the peptide world, make sure you're following me, Kai Rivera, wherever you get your scientific kicks. And of course, keep tabs on Peptides.today for all the latest discoveries. Protein shape is crucial. If folding fails, the protein often cannot perform its intended function. Misfolded proteins can disrupt cellular activity, a key area of study in many research disciplines. Now let's explore enzymes, the speed demons of biological chemistry. They are biological catalysts. A catalyst significantly speeds up a reaction without being consumed, enabling crucial biological processes. Without enzymes, many reactions would proceed too slowly to sustain complex biological systems. Enzymes can accelerate reactions thousands to millions of times faster, a critical rate for life. While most enzymes are proteins, ribozymes, made of RNA, also exhibit catalytic activity, a Nobel-winning discovery. This expanded understanding reshaped how scientists define enzymatic functions and molecular roles. Ribosomes, the molecular factories that build proteins, themselves comprise both RNA and proteins. This demonstrates the profound interconnectedness of biological systems. Proteins build the machines that build more proteins. Now for peptides, the shorter chains of amino acids, often considered under 50 amino acids long. Their smaller size means peptides frequently function as signaling molecules, relaying instructions between cells. Researchers are excited about peptides due to their precise interactions with biological systems. Their small size makes them attractive for development in various research contexts. However, many peptides degrade quickly and may require specialized delivery methods in experimental settings. Peptide research is an incredibly active and evolving field within modern biotechnology, full of potential. Always remember that many discussed research compounds are experimental and require careful scientific oversight. What an incredible journey through the molecular landscape! Understanding these fundamental differences empowers us all, from seasoned scientists to curious autodidacts, to approach research with renewed clarity and excitement. The synergy between peptides, proteins, and enzymes is truly one of science's most captivating stories, constantly revealing new puzzles to piece together. Keep digging for those molecular gems. Follow me, Kai Rivera, for more deep dives, and definitely follow Peptides.today. Before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.